Hello everyone! The reportable disease I chose for this assignment was yellow fever. The main objectives for this presentation are to learn about the background and discovery, characteristics, pathophysiology, transmission of the disease, sign and symptoms, current treatments, grants, and ongoing research for yellow fever. Yellow fever is a zoonotic disease that affects 200,000 people globally and causes on average 60,000 deaths per year. This disease is mainly seen in Africa and parts of South America. The causative agent for this disease is yellow fever virus and is spread mainly through infected mosquitoes and sometimes infected ticks. The organism that carries yellow fever virus will greatly depend on its geographic location. The signs and symptoms will depend on the type of transmission and the severity of the disease. Currently, there is no cure for yellow fever, but there are certain treatments, preventative measures, and movements to stop the spread of yellow fever. Yellow fever originated in Africa and spread through South America due to the slave trade. Historical data showed that yellow fever eventually spread to other parts of the world by European exploration and colonization. U.S. troops during the Spanish-American War were infected with yellow fever and contracted the disease from Aedes aegypti mosquitoes. There is little evidence to show why yellow fever has not spread to other tropical areas of the world, such as Asia. The term yellow and yellow fever refers to the jaundice that some patients may experience when exposed to this disease. 15% of people who get yellow fever are inclined to develop serious illnesses that can result to bleeding, shock, organ failure, and even death. There are 53 species of the Flaviviridae genus. Flavus, meaning yellow in Latin, is known to be the largest among the Flaviviridae family in the microbiome. Flaviviruses can be subdivided into non-vector or known vectors. Vectors for most flaviviruses are known to be tick-borne or mosquito-borne. In this presentation, the main vector for yellow fever is mosquitoes. Most flaviviruses are composed of more than 70 small, positive-sense single-stranded RNA encoded with two multi-domain proteins, NS3 and NS5. The two multi-domain proteins alter the function of the lymphatic system and create a cascade of events in the host cell. Yellow fever virus affects the function of the lymphatic system by altering the protein structure. The lymphatic system is part of our immune system, and its essential function is to protect our bodies from foreign invaders, maintain fluid levels, absorb fats from the digestive tract, and to transport and remove waste products and abnormal cells from the lymph. To put in simpler terms what is happening, the arrangement of RNA genomes and the coding for particular polyproteins and non-structural proteins are altered and allow a cascade of events to occur in the host cell. Many flaviviruses, like yellow fever, enter the host cell by being deposited into the skin's epidermis via mosquito bites. As the flavivirus enters the host cell, it will start to infect other cells in the body, such as keratinocytes and skin dendritic cells, which both play a role in the immune system and are common targets for flaviviruses. Studies that have been conducted on yellow fever focus on the correlation between yellow fever virus and the tropical environmental factors such as temperatures, rainfall patterns, altitude, and mosquito distribution. There are 3 transmission cycles for yellow fever virus: sylvatic, intermediate, and urban. Sylvatic yellow fever, also known as jungle yellow fever, occurs when humans enter jungle areas where the disease can be passed from infected monkeys by wild mosquitoes to humans. Sylvatic yellow fever is considered to be the predominant transmission cycles in the Americas. Intermediate yellow fever is the most common type of yellow fever outbreak and is present in areas where semi-domestic mosquitoes can result to human and or monkeys contracting the disease. Lastly, Aedes aegypti species transmit urban yellow fever to humans without transferring it to other primates. Signs and symptoms of yellow fever will greatly depend on the severity of the disease. When the host cells are infected by yellow fever virus, the infected dendritic cells migrate to the lymphoid organs, circulate into internal organs with replicated virus cells, and causes central nervous system damage. Yellow fever causes flu-like symptoms, which include fever, headache, muscle and joint pain, nausea, vomiting, and rash. Yellow fever virus replicates in the liver, heart, kidneys, and lungs and causes a range of clinical symptoms, such as being asymptomatic and having renal and hepatic failure. With yellow fever, symptoms develop 3 to 6 days and also include high fevers, seizures, encephalitis, paralysis, jaundice, hemorrhage, coma, and eventually death. Yellow fever can be analyzed based on the type of strain. The biosafety level is determined by the strain type. For yellow fever, there are two types. The first one is wild-type yellow fever virus, which is considered to have a biosafety level of 3. The second strain type is the yellow fever virus 17D, which has a biosafety level of 2, which is currently used for treatment for yellow fever in many parts of Africa and the Americas. Currently, there is no cure for yellow fever. However, scientists in research and development are creating a yellow fever vaccine with the yellow fever strain 17D. Live attenuated 17D vaccine was developed in the 1930s and has greatly reduced the number of yellow fever cases in Africa and South America. There are currently 3 17D substrains in production. The first one is 17DD, manufactured in Brazil. The second one is 17D-213, which is manufactured in Russia. And last, 17D-204, manufactured in China, France, Senegal, and USA. Although there is an improvement in a public health perspective, there are few studies to support that live attenuated 17D vaccine is considered to help protect against yellow fever in the long run, and this is due to not knowing the longevity of the vaccine. Some preventative measures for yellow fever include Environmental Protection Agency approved insect repellents, wearing long sleeve shirts and long pants, treat clothing with permethrin, which is an insecticide that kills and repels against mosquitoes, and sleeping in an enclosed area or in a mosquito bed net. I, which stands for Eliminate Yellow Fever Epidemics, is a campaign developed by Gavi, UNICEF, and WHO. The mission of this campaign is to strategize and protect at-risk populations, prevent international spread, and contain outbreaks rapidly from yellow fever. According to WHO, there are 3 critical global agendas to successfully eliminate yellow fever. The first one is international health regulations, second one is global health security agenda, and last is the universal health coverage. To conclude this presentation, studying the environmental factors of yellow fever virus has greatly improved the understanding of where yellow fever is most likely to occur. For any associated flavivirus diseases, it's important to study the relationships between the host, virus, and environment to better create treatments and preventative measures to eliminate health disparities across the world.